So the last way that cells can control gene expression is through post-translational control. In this type of control, cells will often modify proteins, and this can affect their stability or their activity. So several of these we've talked about already, things like ubiquitination when we talked about p53. When p53 is bound to MDM2, MDM2 adds ubiquitination molecules to p53. This causes p53 to be rapidly turned over and destroyed. It causes it to be recognized by certain proteolytic enzymes, and it is just turned over pretty rapidly. When I was a graduate student, there was a girl who was studying p53 in the lab with me, and it was very difficult for her to study because when she tried to stain a gel with this, many different proteins are being ubiquitinated throughout the cell. And so she thought she could just tag the ubiquitin and look at that, but it made it much more difficult because there are hundreds of hundreds of proteins, if not thousands of proteins, that are being turned over through this process. Other ways that that proteins can be regulated is through different modifications. Phosphorylation, glutathione, acetylation, and so on. So in phosphorylation, this is going to be an addition of a phosphate group to that particular protein, sometimes one, sometimes more. Through this will cause cause the protein to possibly react in different ways, possibly causing it to gain functions, possibly causing it to lose functions. When I was working as a postdoc, one of the girls in the lab was studying the phosphorylation ability of the glucocorticoid receptor, and she found that it definitely changed its activity when she caused mutations so that it no longer could be phosphorylated at certain locations. Glutathione is another form of modification that can happen. Acetylation is a modification that can happen to things like histones and so on. So in conclusion, this week, make sure you're catching up on the Mastering Genetics homework if you've started to fall behind. Same with the discussion forums. I know several people haven't been commenting lately. You can catch up on that. Try to do that as soon as you can. Also, continue to work on your student project. There is a quiz this week. And lastly, hang in there. We are almost towards the end of the semester. There's just a few more chapters that we're really going to be covering. So hang in there, and I hope you've enjoyed this semester so far.